Please welcome Dr. Ilya Finkelstein and Dr. Stephen Ekker from the University of Texas at Austin.
Thanks, Jay. Thanks to the team at Austin Forum for putting this together and inviting us out. I'll do the first half, and Steve will come in and tell us a little bit about his work. And I want to thank all of you for coming here after work to listen on this topic. This is going to be fun. So today we'll talk about gene editing, a technology that, whether you know it or not, has already touched your lives. You've probably already consumed gene-edited food, whether you know it or not. Today I want to tell you a little bit about how this technology started, where it's going, and then we'll discuss potential future ramifications, both in terms of technological development and the promise of this new technology, and also what it means for society. All right, so here you see a picture of Darwin's finches. These are the finches that he sketched in his notebook. This is what he looked like at the time when he was working on The Origin of Species. And he noticed that these finches adapt to their niche, right? They all come from the same progenitor bird, but they've adapted to their niche, especially in their beak, to feed at different ecologies, right? And so he wrote in his notebook, slightly abridged, seeing the diversity of structure in one small, intimately related group of birds, one might really fancy that a species has been taken and modified for different ends. And this is where life has stood for, essentially hundreds of thousands of years. Natural selection and evolution has driven the development of species and the developments of features of different subspecies. Fast forward to about a few years ago, and I'm just showing you some headlines, all right? Scientists can design better babies, should they? A new technique for manipulating genes holds great promise, but rules are needed to govern its use. This is all a couple of years ago. And perhaps most strikingly, in 2018, first CRISPR babies, 6 questions that remain. I think there's probably more than 6 questions in all of our minds about these CRISPR babies. So the point I'm trying to make now is, instead of waiting for thousands of years of evolution to shape our features, we now have the ability for the first time of all living things on Earth, not only to shape our own genetic future, but to shape the genetic future of all living things around us. And we are doing this very rapidly right now. So today we'll talk a little bit about how that works and what that means going forward. So this technology, as you can imagine, holds great promise, but there's also lots of dangers and unresolved questions ahead. And I want to leave plenty of time for discussion, so I'm going to try to keep this relatively brief, and I'm sure you all have a lot of questions that we'll address together with Steve together. A brief timeline. This is one of the fastest technologies to be commercialized, from basic laboratory discovery, fundamental science, to clinic and beyond. Okay, let me just give you a quick rundown of where we are now. The first evidence of gene editing using a technology called CRISPR, which I will describe in a slide or two shortly, happened in 2013 in human cells in tissue culture, which means in a plastic dish. Shortly thereafter, pigs that were resistant to swine flu were created. This is very important for agriculture, as you can imagine, because swine flu claims a lot of the herd, a lot of the pigs. And there's no good vaccines for pigs. In 2017, a company developed pigs that were good, suitable donors for human organ transplants, setting up a possibility to solve the acute organ crisis that plagues human health. In 2017, the first viable edited human embryo was described in the United States. The first human clinical trials were started in 2018. The first engineered humans were born in 2021 in China. If that doesn't send goosebumps down your back, I don't know what will. Okay, so this is a crazy side story we can get into. Essentially, this was a quote unquote rogue scientist in China who conducted these trials using quote unquote donated eggs and IVF. And the purported goal was to create children that were HIV resistant. So there's a receptor that the HIV virus must bind to enter your cells, and this receptor was erased. Again, don't know. There is no follow-up, but these girls were born. So there are viable children who have been gene edited now, and they will inherit and they will transmit these mutations, whatever they have done to them. This is very secretive right now. They will transmit it to their progeny. Okay, and then getting back to US, the first gene editing treatment for sickle cell disease is now available in the clinic. Okay, so there are now many such treatments in the clinic. Now, so let's see. Here are these cute little pigs that are going to be hopefully solving the future organ shortage. And here are a few other implications for gene editing beyond human use. Okay, so gene editing has rapidly entered the human food chain because it accelerates the selection of positive traits and the creation of new traits that can't be created by breeding or inbreeding by traditional methods. For example, in 2014, just a year or two after the discovery of this fundamental technology, people were already developing disease-resistant wheat, herbicide-resistant canola, which has now entered our oil supply for foods. And by 2022, I looked this up, there's now over 40 gene-edited, engineered crops in the food supply across the world. Here are some mushrooms you can go buy. They won't be edited, they won't be labeled as gene-edited, but they have a mutation in them that erases a gene causing browning. So these mushrooms stay white longer. So the food supply has already received this gene editing technology. And so I keep saying the word CRISPR, it's an acronym. The acronym is scientific set of terms, which isn't relevant here, but I want to tell you a little bit about where this comes from. And there's an important message here. This comes from fundamental research with no goal other than curiosity. Okay. This thing that's revolutionizing, this discovery that's revolutionizing all of biomedicine right now came from fundamental curiosity-driven research. So this is one take-home message I want you to remember. Your taxes, thank you, support fundamental research with a long tail of potential benefits that we won't know for decades. So it all started in 1989, when the humble E. coli bacterial genome was finally decoded. The genome is the operating system of the cell. It consists of 4 letters, A, C, T, and G. And at that time, the technology was progressing far enough that people could start decoding partial parts of the bacterial genome. Now, E. coli is a model system in science, and people have studied it for many decades. At that time, in this very obscure paper at the time from 1989, a group published a curious observation with no known explanation. They noticed that if you just put the letters of the E. coli genome, there appeared to be this weird set of letters that kept repeating, like a stutter or a copy-paste mistake. It kept repeating over and over again, and then there were some like random letters over here, and then this pattern repeated, again, repeated, again, repeated. They described this as, in technical terms, an unusual structure was found. That was the state of the field in 1989. And there it lay for decades. Okay, briefly to summarize this in a schematic, you have this repeat in black, then you have something called a spacer in red, then another repeat, another spacer, another repeat, and so on. This can go on and on. Okay, so they didn't know what these spacers were, okay, but they thought maybe these spacers, these random pieces of DNA, are some sort of historical archaeological record, if you will, a fossil of invasion of other DNA pieces into the genome. And that was really quite amazing that they could guess that. There was no real evidence. They could guess that. All right, that's where it lay for decades. It was just described in an obscure paper. That's it. The story takes a wild turn. Now we're going to talk about yogurt. Okay, so this is yogurt. The DuPont company owns the brand Danisco, which makes the yogurt Yoplait, which you may have enjoyed yourself this afternoon, or maybe last week, or whatever. And when you look at yogurt, you see this. When I look at yogurt, I see this. This is lactose acidophilus. It's responsible for taking all those milk sugars and converting them into delicious sour yogurt. Now, when DuPont makes massive tubs of this Yoplait yogurt, they sometimes have a problem. For reasons that are pretty clear, some of their massive production vats fail, and they have to scrap the whole process and start over again. Scrub, clean it up, start again. Very costly process that takes the whole factory down. Okay, so what is this process? Well, if you take an electron microscope, which can do very high magnification, you can see that sometimes these lactose acidophilus bacteria have a bad day. You see these little things sticking off of them? Well, guess what? Bacteria can also get a viral infection. So when bacteria get a viral infection, the viruses use that bacterial host to make more copies of themselves. Here's a blow up. Here's a little virus sitting on top of a bacterium. Okay? The virus takes over the bacterium, makes copies of itself, and then explodes the bacterium. That causes more viruses to come out, more bacteria get infected, your yogurt production fails. Problem. But not all vats were susceptible to this viral infection. Some vats continue to produce yogurt, and the bacteria seem to be resistant to the virus. It wasn't that the virus wasn't there, it's that the bacteria were somehow immune from that virus. Okay? Not only were they immune from that virus, but they could actually adapt to new viruses that were infecting the rest of the culture. So Yoplait had a very strong business interest in figuring out what was going on and making sure they can expand that to all of their production facilities. Briefly, quick biology lesson. When a phage, which is the technical term for a bacteria that invades, a virus that invades a bacterium, when a virus attaches to the bacterium, shown here as a little red circle, it injects its genome, its DNA, into this bacterium, makes more copies of itself. Eventually, the bacterium literally explodes, bursts open, and then more copies of this virus come out and then infect more bacteria, and the process repeats. But something was stopping the process right here, right? These bacteria learned to be immune to these viruses and thereby stop this whole cycle. And that's where CRISPR comes in. So far, it's the only known adaptive bacterial immune system. So bacteria, like us, have an adaptive immune system. And the protein CRISPR, Cas9, for example, is really the driving force that allows bacteria to survive these viral infections and adapt and learn which viruses are in their environment. I'm going to go through the biology in 1/2 slide. If this is not interesting, skip it. I'll give you a take-home message in a second. So, as I was mentioning, here's a blow-up of what's happening when a virus infects a bacterium. The virus literally pokes a hole through the bacterium, injects its DNA into the bacterium. Now, the bacterial immune system can actually pick up pieces of this DNA and insert those pieces of the DNA into its own genome. And that's kind of like a molecular recorder. It's a recording device that says, oh, this piece of DNA is foreign. And that's exactly the spacer that I referred to earlier. Now, these spacers are put between these special repeats. That's why these repeats are important, because they mark places where an external DNA entered the bacterium, a bad DNA. Then this DNA gets converted into RNA, which isn't particularly important, has some special properties, and a protein picks up this RNA, which is complementary to this DNA. Now, this protein is like a molecular scissors. But these scissors only work if they're targeted, programmed, if you will, to attack that viral DNA. So then, in a process called interference, this Cas9 protein or CRISPR-Cas9 protein will go up here, attach itself to the incoming viral DNA, and then cut it up. So the take-home message of this slide is that if you have this protein Cas9 and you suitably program it with a homing signal, or maybe consider it a loaded gun, with this being the bullet, this protein will go and find whatever piece of DNA that's complementary to this red piece of this program and go and cut it precisely. That's how these very dangerous weapons don't kill the bacterium itself, they only kill the virus. So this is the adaptive immune system known as CRISPR. And very quickly, people figured out that you don't have to just study it in the context of bacterial viral interactions. You can actually take these proteins and transplant them into any organism and use them as a precision sort of homing scalpel to cut the genome of any organism you wish, including our own. And so that's where we are today. Skipping over all the boring work and billions of dollars of research, we now have the ability to take Cas9 or related proteins. There's now a universe of such proteins, and Dr. Ekker will tell us about how AI is transforming that, and then put them into any kind of organism, including ourselves, target a specific gene or maybe between genes, and cut it. And with suitable experiments, that's called a gene knockout. That's deleting a gene. Maybe the gene causes a disease. With specific enhancements, we can now do gene replacement. So maybe a cut and paste operation, or one hopes in the future, chromosome inactivation for various trisomies, like XXX and so on. So that's where the technology grew from really fundamental studies of, wow, that's curious, to, oh, my yogurt culture isn't dying. How can I make that better? To, oh my gosh, we can edit anything anywhere. This is the value of fundamental research. All right, so now that we are able to edit human cells and those of our companion animals or our food supply, etc., I'll just give you one slide on technical hurdles, because there are many, many, many technical hurdles. Well, first of all, I said it's a precision scalpel, but everything has a tolerance, right? There is nothing that is 100% precise. And recall that these systems have evolved, there's that word, they've evolved to fight very small viruses. So here's an order of magnitude estimate of the genome size. How many letters are in the genetic program of a virus that infects a bacterium? You know, maybe 10 to 100,000 letters in that genome. That's a virus here. And so that's fine. So the scalpel only has to be accurate in about maybe 1 in 10,000 or 1 in 100,000. E. coli has that bacterium has a 4 million base pair genome. Fruit fly, another common model organism in science, 100 million base pairs. We're getting to 3 billion base pairs for a human. And if you want to edit plants, 150 billion base pairs. Plants have massive genomes, much, much bigger than human. So if a tool is accurate on this scale, how accurate is it on this scale? Because the stakes are high. Unanticipated edits can themselves be lethal. They can cause various oncogenic cancer-causing mutations. And that has happened in the clinic or in clinical trials. All right, so that's one problem. How do we get unanticipated off-target editing down to a minimum? And there are now many solutions to this. We can engineer the enzymes to be more precise. We can use machine learning and other technologies to select the precise targets better, so we can reduce off targets. And we can verify the edits by other means. All right, another technical problem. How do you deliver this technology to cells? This is a form of protein therapy. And I think Dr. Ekker will touch on cell therapies. Now we have a new class of therapies called living cell therapies that are enabled by this technology. Again, complicated slide. I'll take you through it briefly. You don't have to remember all this acronym soup. This is from a technical report. But we need to be able to deliver it to cells. And delivery is really, really challenging for this kind of therapy. It's not like you pop a pill, it goes in your stomach and gets into your bloodstream. No, you have to deliver it somehow. So adeno-associated viruses. So people have used viruses for delivery. Viruses have their own challenges, including inducing a strong immune response. You can try to micro-inject it to the site. This works in the eye and in some other areas, but it doesn't really work globally or systemically. One way that seems to be fairly far along is using lipid nanoparticles. This is the same technology and mRNA delivery. This is the same technology that was scaled massively globally during COVID. So one of the very few silver linings of the COVID response was that we were able to scale RNA therapies globally and show that they validate them, show that they work. And these therapies, these delivery methods are useful for delivering gene editors as well. Other problems include unanticipated immune response. So your immune system, not the bacterial immune system, your immune system recognizes these proteins as foreign. You can have an unanticipated response. Also, all of the delivery technologies bring their own immune challenges. So they could themselves cause immune responses. That's why when you get that COVID shot, you feel pretty crummy the next day because you have an immune response against the lipid nanoparticle. And another major problem is cost. Let's talk about that because that's a societal challenge that we have to address. All right. So aside, let's meet Victoria Gray. Victoria Gray is the first patient to be cured, keyword cured, from sickle cell disease. She was treated with CRISPR. There was no cure for sickle cell disease. It's a disease of hemoglobin. It's a blood disease. It causes the blood cells to malform. They can't get through the vasculature. They get stuck in the capillaries. It feels like you're being stabbed with 1,000 knives all over your body. Prior to that, the only way to deal with this is management, blood transfusions, incredibly difficult. First person to be treated with sickle cell disease. Sickle cell disease is now one of the first FDA-approved clinical therapies for CRISPR gene editing clinical therapies for in the world, and specifically for sickle cell disease. All right, this is still an arduous journey. Okay, it's a cure, but it's an arduous cure. So what has to happen for this to work is that special hematopoietic stem cells, so stem cells that can make blood, are extracted from the bone marrow. The patient's bone marrow is wiped out in a very invasive procedure. The cells are edited ex vivo, means they're taken out of the body, edited in a petri dish, checked, double-checked, triple-checked, and then reintroduced into the patient, where hopefully they recolonize the bone marrow and replace the blood supply with a cured blood. Okay, this is now called Casgevy, or I don't know how to pronounce it, but the price tag is $2.2 million. Okay, $2.2 million. With this price tag, even though it's a cure, remember it's a one-shot deal, right? One-shot deal, cured. That price tag, despite being a world leader in this, the company that developed this technology is actually unable to sustain the business model because a cure is an unsustainable business model, right? A chronic patient is a sustainable business model. A cure is not a sustainable business model. So we have to think about that too. Complicated table. I pulled it out just so you can see where we are today. So these are the therapeutics that are now in the clinic. These are just 12 out of 27 therapeutics that are today in the clinic using this CRISPR bacterial system, this bacterial immune system to cure diseases in humans. You can see some of them are ex vivo. Most of them are ex vivo, means the cells have to be extracted from the patient, cultured, edited, tested, and then reinserted back into the patient. And here's the price tag, right? So for in vivo therapies, we're hovering around $2 to $3 million for a single shot. For ex vivo therapies, most ex vivo therapies, we're targeting about the companies are targeting about $500,000 per shot, but these are not single cures. I should note that a lot of these are what are called CAR T cell therapies, and this is the living cell therapies. I don't know if Dr. Ekker will touch on this, but these kinds of therapies require the patient's immune cells to be extracted and edited and reinserted, and they've been remarkable against blood cancers of various types. Really amazing remission in blood cancers using a technology that was enabled by basic science driven by curiosity. However, the price tag is high. As a society, we have to grapple with this. This is truly therapy that is developed for the patient. It is a one-off therapy. There will not be, it's not like the computer world where you make 1 billion widgets and it gets cheaper. This is tailored to you from your own cells. So the cost of goods is very, very high, and I don't think there's going to be economies of scale here. So can we afford the therapies? Who's going to bear the cost? Another societal challenge. All right, so I'm going to wrap it up here. I'll give you my future outlook. I did this about 5 years ago, and I was not super off, but I'm going to try this again. Don't take this, take all of this with a grain of salt, because if I could predict the future, I'd be a richer man. So where are we going with all of this? All right, so I firmly believe that gene editing is an interim solution. Initially, biology was rocked by the genome read revolution. The ability to sequence genomes very cheaply changed biology about 20 years ago. The Human Genome Project was really the culmination of that. Now we're in the genome editing revolution, some 20 years later. And I think 20 years from now, we will be in the genome writing revolution. What do I mean by that? Nowadays, people in my laboratory can just order a piece of DNA, type it into a text box in a web browser, and 2 days later, a FedEx envelope shows up in our lab with that piece of DNA in a test tube. We can do this for whole genomes. We don't have to do this for short pieces of DNA. This has already been demonstrated for viruses, for fully synthetic bacteria, and for fully synthetic yeast. So this is Baker's yeast. People in an international consortium, you can see all the participating institutions, have been able to synthetically, completely, chemically make a whole genome. These are all the different chromosomes shown here, and this shows which chromosomes were completely made and which chromosomes were partially made. And that yeast booted up. You can actually write a genome, stick it into an anucleated cell with no DNA in it, and the cell wakes up and lives. So we can program life. So the Human Genome Project, how far away, a genome writing project, how far away are we to making a synthetic human genome? I think the technology is completely there, 100%. It's a cost challenge. As soon as we decide to spend sufficient funds, we will make a synthetic human genome. And you can imagine what that means. We can write, we can edit out, not by CRISPR, but by just programming into a text box, every possible disease variant. We can increase other features. We'll talk about that. All right, chromosome or genome transplants. All right, so if we can make a synthetic genome, wouldn't it be lovely if we can make synthetic genomes enter germline cells to perhaps create de novo humans with completely synthetic DNA? Related, there's a company in Texas that is seriously trying to use this for de-extinction of related species. What do I mean by de-extinction? They're working on bringing back the woolly mammoth. The idea is they have sequencing from woolly mammoth fossils. They know exactly the sequence of the woolly mammoth. Taking a modern elephant, it doesn't take that much editing to convert a modern elephant cell into a woolly mammoth. And then using in vitro fertilization in modern elephants, a woolly mammoth would be born. So de-extinction can be a really interesting opportunity to claim back some of the biodiversity that we're losing today. And then I won't talk about this at the moment, but this technology can also be used to collapse food supplies by forcing, and collapse species by forcing potentially underperforming genes or killer genes into the population and forcing them to spread. These are called gene drives. They've been demonstrated in the lab and they've been demonstrated in the field. People talk about gene drives in the context of eradicating mosquitoes. And I have a whole spiel on that, but we won't get into it today. But of course, equally, gene drives can be used to eradicate valuable species. And state actors and non-state actors are certainly working on this. So just to give you my one last slide before Dr. Ekker steps up, just so you know that genome transplantation isn't a wild dream. Some of you may be aware the genome transplantation of the mitochondrial genome. The mitochondria is a separate part of your cell that gives you all the energy, all of your ATP. Mitochondrial genome transfer has already been done. So there are now 3 parent babies out there that have mitochondria from a donor, their maternal DNA, and their paternal DNA. So three parent babies are approved as a therapy in the United Kingdom, where a mitochondrial genome is literally transplanted by a micropipetting technique where you jam it into the egg and put the new DNA into the egg. And can we do this for the whole nuclear DNA, and why not? I think technically there's no hurdle. It's a cost hurdle, but it brings up a whole raft of questions about taking our own genetic fate from that of evolution. I'm going to stop there and let Dr. Ekker take over.
All right. Okay. So thank you very much. Really enjoyed that, Ilya, and thank you for the opportunity to meet you. And it's my first forum. I started exactly a year ago at the University of Texas at Austin. I just want to ask a question. How many of you are engineers in this room? Okay, great. I'm going to start. This whole section is for you. Okay. All right. So in 1978, this was the Apple II. It was not an accident that there was a scientist named David Brin, Dr. David Brin. He was at NASA, and he had access to a word processor. That was a key piece. And Dr. David Brin asked himself, what if you could code DNA like you can text on a word processor? And what would you do? Okay, so if you know David Brin as a novelist, I want you to not give me an answer because we're going to go to the answer at the end. But for the rest of you who don't know this, we're going to go through this story. Okay, so let's start with what are you going to do with it? Are we going to talk about therapies? We talked about that. Are we going to do bioweapons? Okay, science fiction. Maybe it's both, like Captain America. Is he a weapon or is he a therapy? What is he? These are the ideas that come out of science fiction. And I just want to tell you the true story that science fiction is deeply inspirational. Many of the things we do first came out of people like David Brin, who are they remove some sort of technical barrier and say, well, what if I could, what could you do? Okay, so Ilya talked a little bit about the therapy, but keep in mind that what are you doing with gene editing is you are editing the operating system of the cell. What I think most people misunderstand is that we're not one cell. We're actually made out of 37, on average, trillion cells, where each cell has a flash drive. I was going to say hard drive, but that's because I'm too old. A flash drive that you have to make changes to. So, and each one of those is per cell. So if you want to do a major change in an adult, this is the challenge. And this is the bottleneck. But actually, our human body is also amazing. Okay. And in fact, we heard about it. You can actually pull out portions of this, make the changes, put them back. That's how you got the sickle cell disease. Muscular dystrophy is being cured because our muscle is a syncytium, and you can actually hit more than one region because of the syncytium. And CAR T cells. So CAR T cells is basically pulling out your immune system, reprogramming it to target a cancer, and you put it back in, and those cells go through, and they will actually remove the cancer entirely from the body. It's a programming. So there's amazing things that can be done. Okay, so so my journey. Why am I here, and why am I talking to engineers, and why does it seem great? Okay, I am actually a trained semiconductor physics. I was doing gallium arsenide semiconductor designs in the 1980s. And I had the opportunity and the luxury to work in Dr. Carl Wose's lab at the University of Illinois and Urbana-Champaign. At the time, he was doing early sequencing to understand life and understanding how to interpret that read stage. And he convinced me. He actually said to me, Steve, why spend the rest of your life coding computers when you could code life? So I've been a genome engineer since the mid-1980s, okay? It was really hard when I started. There was very little you could do. My entire PhD dissertation was trying to understand how you could just make and do even an individual protein to spot in a single part of the genome. It's like a cursor, just like figure out how do you want to do. But that's the whole story. I want to tell you the story. See if I can get this to go. Because this is my disclosure slide. These are a series of my companies that I participated in or worked with, and they all have this core premise, which is how you can code life for innovation and impact. And I'm just going to talk briefly about a couple of them so you understand that what do I mean. Okay, so these two companies are human therapeutic companies. And in one, you're trying to take B cells, the cells that normally make antibodies, you pull them out, you code them to make therapeutics, and you put them back in the patient. And last week, they actually reported the 9-month treatment of the first patient, where that patient's complaints are now, I have to go in once a week for my therapy. So his life has returned to normal, his full remission for a rare disease called MPS. Okay. That took about 20 years to develop that technology for the first patient, but it's exciting because the first patient is really showing. an ongoing company we're developing is called Primera Therapeutics. It's actually to edit the mitochondrial genome. We're very early, but those are two stories that are ongoing. As of a few hours ago, I can actually tell you the story that for CAR T cells, this is Ilya Laboratories, building cures for dogs as a primary. Put the first 2 dogs in remission with CAR T cells. That's the first ever. What's important is it's using non-virus approaches, and the cost of goods is $500, not $500,000, okay? And so the possibility of really cracking the space because you've innovated on how you build and do this is the possibilities. Okay. And then what else can you do with this technology, right? We talked about therapies, but there's other things you can do. Friends of mine, and I'm actually a shareholder, called Recombinetics, they actually are fighting climate change by making, for example, heat-tolerant Angus beef. And those are being grown in Brazil and Argentina and allows that to go. I'm not going to talk about whether you want to be vegans or the climate, but the answer is that you can also take it. We actually started a company called Forge Azul to grow a form of seaweed that would be salt tolerant so that you can go to higher density so that you can use that to replace a lot of petroleum products. So these are all using gene editing. These are all technologies for that space. Okay, so I want to start with David Brin. What if you could code DNA like you can to text on a word processor? And although he talked a little bit about, editing people, he also asked a question, what are your answers? I had the opportunity to have David give a keynote talk at the Genome Writers Guild. So my question for you is, what would be your answers? You are engineers. You can now code life. What would you do, right? You have a couple of people that want to give some thoughts? What would you do if you could actually code DNA from the operating system of the cell? Yes, John. Okay, so you do an organism that would consume free plastics. Okay, that's great. What's that? Perfect eyesight. So people. Okay, so those are all cool things. Okay, what I'm going to tell you is that he actually did something I think was amazingly different than all that. He actually imagined giving the ability to talk and give tools to dolphins. Okay, and this is important. David Brin was a NASA scientist, and he looked at the human body and said, we're terrible in outer space. We actually should live on gravity, right? With gravity, and we're actually vertical. And when you're out in space, you're actually really, right? There are really amazing, intelligent animals on the planet that actually would be better astronauts, and that would be fish, right? Or dolphins. And he he realized, well, if the DNA encodes the ability to talk, why not just add talking to the dolphins? And so the whole story is about the launch of the first interstellar spaceship crewed by the dolphins, okay? He went further and he said, well, if that's a principle in a galaxy, what if you end up actually having, that's how one species becomes intelligent, is that one other species actually engineers that species with this ability to think and talk, and he invented the concept of uplift. So he actually invented an entire universe around gene editing and this core principle. I'm not creative. David Brin is creative, right? But he started because in the 1970s, he had access to a word processor and this amazing imagination. Okay, now I have to give a little personal because this was very important to me. In the summer of 2012, I'm reading a prequel to Star Tide Rising. I read this, it's a Hugo Nebula book I wrote when I was younger. And I had to put the book down. And for weeks, I couldn't pick it back up. It's a true story because we were about to make this public, this Nature paper in 2012. We published the ability to change the germline of vertebrate animals to single nucleotide resolution with a technology in 2012. We had made, but David Brin's idea possible in reality. And so it's very interesting. You went from science fiction to reality in one scientific lifetime. So it's interesting. So that's the life. What's next? I don't have a great crystal ball, but I just want to offer that for those of you in the room, you know that the majority of computers, right, on the planet, right, like the mobile phones, the Linux, the servers all run Linux, right? So it's a hardware and software. Well, until this summer, we were always dealing with proprietary software for gene editing. But what actually happened with the AI revolution was a company, there's actually more, but Profluent actually used the AI generative biology space to actually make a Linux for CRISPR. And it's a Cas9 system that's fully open source. And I was supposed to bring a tube to Ilya tonight for him to use. And so we are entering a space where it is as open a playing field as possible, where there is no constraints on the toolbox. It's all a constraint on what do you want to do with these technologies from a possible. And I believe that we have a huge, amazing future ahead. So with that, I'm going to hand it back to Ilya, and love to have a conversation afterwards.
Thanks, Steve. Don't go far. I think we'll just drag 2 chairs on in a minute. But if you want to drag on for a little discussion so we can both answer questions. But since you segued so brilliantly into another topic, I think it's worth touching on, because Steve said we can do a lot and the tools are becoming available and democratized, where now you can do this in a home lab even. So that brings me to, if you humor me, just give me a few more minutes, the ethical and societal implications of where this is going. So are we playing God, right? Yes. Let's see. So let's talk very, very quickly about something that deserves a whole course in and of itself, but I'll just do it in 3 slides. All right, ethics policy and CRISPR-based technology. All right, so I'm not going to talk, there's 2 technologies when people discuss the ethics and societal impact of gene editing. We've started this one. This CRISPR offering a very powerful technique that really supersedes almost all prior methodologies that were very, very difficult back in the early 2000s. Their second one is these gene drives, sometimes referred to as gene bombs, the ability to change a whole species, not at an individual level, but across the whole niche, ecological niche. This is supramendelian inheritance and the rapid spread of a gene or set of genes in a population in the wild. Again, I won't talk about this, won't have time on it for it, but it's a sort of a fascinating and exciting area. All right, so let's talk about the ethics of gene editing. Right? First and foremost, we need to make a distinction between editing the germline and the somatic gene editing. So somatic gene editing is where we are with current therapies, where you edit an adult and that information is not heritable, meaning you won't pass it down to your progeny. For example, if you're cured of sickle cell disease, they would have to be cured separately. Now, if you edit the germline, then you have CRISPR babies. The kids that were born to that Chinese rogue scientists will then go on to have children that will continue to have those mutations. So now you're really starting to change the human population and ultimately probably lead to speciation. Because if they are truly HIV resistant, why would they ever mate with us, right? Just a thought. So there you quickly get into the space of positive eugenics, the selection of positive traits. So let's talk about that briefly. Let's start with somatic gene editing, the gene editing that's therapeutically in the clinic right now. So if we accept the modern definition of medicine, which is, to reduce harm and suffering from maladies, then in principle, there's no ethical problem with somatic gene editing, right? The only problems are technical, safety, efficacy, cost, etc. If you can cure a disease by gene editing, you cure a disease. Now, gene editing has had massive setbacks in this space. Safety and efficacy are not minor concerns. Okay, so for example, in 2007, early attempts at gene editing before CRISPR caused leukemia in a number of patients. okay? They were being cured for something else. So yes, you can induce cancers by gene editing. Absolutely. So that's a major problem. But in terms of the societal impact, ethically, to me, it seems reasonable. Now, you guys can disagree, and we'll talk about that. Now, let's talk about editing the germline, CRISPR babies. This has already happened. It may already be happening in a non-government lab or in some secret laboratory in the human population. It's definitely happened in companion animals. All right, so here we have the burden of history. Even in the United States, eugenics was fairly popular not 100 years ago. In fact, here are these eugenic certificates that people would collect for, meeting the right mate. So a lot of dating apps have sprung around this idea of selection by sort of self-sorting. Forced sterilization was quite popular in the United States. California led the way, surprisingly, here. So we have a history burden to consider when we think about positive eugenics to select for traits. Again, when we talk about eugenics, I want to make a distinction between two different kinds of eugenics, negative versus positive. So negative eugenics, you start getting into this gray area. All right, what about being able to remove undesirable mutations or genes from the population? Okay, so for example, disease-causing mutations. Example, cystic fibrosis. It's caused by a single gene. The origin is known. It's very easy to find that gene and test for what kind of mutations cause cystic fibrosis. It causes lifelong suffering. There are ways to manage it for certain subclasses of patients. But what if we could edit an egg or an embryo before it is born so that there will no longer be any cystic fibrosis, no suffering from cystic fibrosis, Duchenne muscular dystrophy, any number of diseases I can list, the list goes on, where you have very clear genetic signature that you can test for, edit, and then have this essentially disappear from the human population. Why not? Is that our medical directive? Maybe it should be. Just some food for thought. I will not give you any answers here, just questions. All right, what about positive eugenics? Let's go there. Why not, right? So here we have raise your hands if you've seen the movie Gattaca. If you haven't, do. It's an amazing movie because it got so much right. Another example, as Steve mentioned, of science fiction inspiring science fact. So there's a famous scene of a pianist who throws his glove into the audience, and they count the fingers on the glove and realize that he was born to play that concerto and no one else can play it. He was engineered for the concerto, right? So should we select for polydactyly? We know the genes. Should we create other positive traits? There's a gene that allows people to live on 5 hours a day of sleep. Who wouldn't want to have a few extra hours of sleep in their day? Doesn't seem to have any other problems. What about a gene that makes you less susceptible to Alzheimer's? We know those genes. We know the mutations that cause them. Okay, so let's think about that. And of course, other positive eugenic traits. Super soldiers come up often in these conversations. Now, the truth of the matter is biology makes positive eugenics very difficult. This is me bursting the bubble. In some cases, it may be clear, but in other cases, it's very hard because of two concepts, epistasis and pleiotropy. What that really means in fancy speak is genes or proteins that are encoded by these genes, these are not just isolated units working in this massive machine. It turns out that you think of these systems, you think of all of these components in your body as a very dense network with many connections and nodes. So changing something in the DNA that maybe gives you blue eyes because you really love blue eyes may also cause disease of the lung and may also cause other problems. How genes interact with their environment and how genes interact with different parts of the body is a very complicated, unsolved problem. And so making a change here, you may screw up the whole network. So realistically, safe positive eugenics is a very challenging problem. And so when the CRISPR babies were born, the outcry was, yes, ostensibly, the researchers' goal was to reduce their ability to get HIV infections, to be resistant to HIV, but maybe making a mutation in that particular gene may have unknown consequences for their health. And of course, the babies had no say in it. They didn't select this. They were forced into this. So positive eugenics is challenging ethically, but it is coming, and it has already, it will happen again. All right, so I'll leave you with this question. The core problem is who decides? And here I want to be humble and remind you that it shouldn't be us, the scientists, who decide, although we have made these dual-use technologies. They are truly dual-use technologies. It should be society that decides. And again, we should remember that different societies have different conceptions of morality and ethics and what it means to be human. What we may find abhorrent here could be quite the norm elsewhere in the world. So different societies will have to come to their own conclusions about what it means to be human and what it means to be able to take our own genetic future into our hands. So with that, I'm going to stop and leave some time for questions.
All right. I'd like to introduce our guest moderator tonight. He is a longtime friend of mine, decades of history together, doing community service and talking about technology and doing fun things. So I'm very pleased to introduce my longtime friend, also a co-founder of BioAustin and part of the Austin Innovation Group, Scott Collins. He will moderate the Q&A session, selecting some of your questions out of Slack. And Ilya and Steve, if you could also come to the front, we'll do all the questions right over here.
Hello, hello.
There were a lot of great questions, kind of from a lot of different perspectives. So good audience tonight. The first one from Ira Berg: is there a test or other way to tell if a human or other organism is a product of gene editing, and what are the implications for selective negative discrimination?
The answer is no. You can't tell because it's DNA is DNA, right? So there's no, the DNA you change is the same chemical core as the DNA you start with. So what's the second question?
Oh, that was basically it.
I'll nuance that a little bit. The US government is very, very actively trying to develop technologies that will make quote unquote right protected organisms, organisms that cannot be gene edited, or technologies to shut down gene editing that is unmitigated. In fact, that is being pursued by some of the most advanced funding agencies that do sort of skunkworks, future-facing research out there. And yes, they're absolutely trying to find signatures, genetic scars, remnants of gene editing, so you could tell in principle, although the modifications are complete if done carefully and completely silent, but maybe there's some leftover of the machinery that did it. So yes, people are very interested from a biothreat perspective.
And part of that also, I understand there's some IP issues with genetic editing and what can come through on that. So that might be interesting to talk about at some point. The next question from James Foster is: do you foresee AI technology fundamentally changing CRISPR science in the near future, and in what ways?
Yeah, so I gave one example, which is to make the toolbox more available and accessible. What's exciting is the next stage, which is really making most of the toolbox we use, almost all the toolbox we use, is inspired by nature. You heard about CRISPR. I did a lot of work with what are called zinc finger nucleases, and I work with tail. These are all taking inspiration from nature, building them into tools. What's exciting is the AI generative biology space allows you to think about, say, a chemical reaction or an outcome, and then making a protein de novo that will, or like an enzyme that makes it, or an enzyme that, and so we're entering this the field from being nature inspired to problem inspired. And that's going to be transformative. It's going to be a few years before this becomes the norm, but it's clearly the future. And so it's not a matter of when whether that future arrives, it's a matter of when and which areas. And there's going to be motivations and enzymes for biosynthesis. Those are going to come first. There's already people doing versions of it, but there's going to be the ability to make things out of biology that we normally synthesize via standard industrial processes. So those are the exciting things. In the gene editing space, there's going to be many new tools that are coming available because of that operations. I'll let Ilya, I'm sure you have other.
Okay, next question, and I think this is important for kind of looking at the industry going forward from Mike Panoff. What discoveries are necessary to reduce the cost of therapies by a few orders of magnitude?
Yeah, so it's partially an R&D question, meaning you need to spend some money to make the therapies cheaper. I'm going to say something that I've said publicly, and I guarantee you doesn't represent the views of the University of Texas. I'm just going to say Steve's views. Gene therapy and gene editing has been subject to regulatory capture in the environmental space tied to the FDA. And it's a very significant issue. Actually, NIST is actually partnering to try to actually release right now, if a company builds a product, the next company, what you would want is the next company to learn from all that work, not replicate all of the prior experiments and only replicate the critical safety experiments so that the second gen and the third gen is cheaper. Right now, it's actually guaranteed to be more expensive because you have to do everything that the prior group did, and then you have to do more special tied to it. So there's a major fundamental issue of regulatory for that. And then there's a second side, which is that we have an asymmetry in the regulatory path. We don't want to have any issues. We don't have any risks. But I do work with rare disease patients, and there's a risk to doing nothing, right? To blocking it. And that's called death. And you actually have many people that are dying from a failure to get the actual therapies into the clinic because of a regulatory burden in the process. And so I sound terrible, but it's not. This is not a political statement. I'm not telling. This is me looking at a clinical view at a regulatory path that's preventing people from actually having treatments in the clinic. From a financial point of view, there's a second, but so that leads to a major issue of big cost for what needs to get done. But I actually have just come to the conclusion that the regulatory burden is a massive bottleneck for being able to bring these technologies forward. The technology is moving forward. I'll give you some of these technologies that are 20 years old. They could be brought forward, but the regulatory burden prevents them from becoming viable. And so these are some of these questions. I would start with that. I'll let Ilya answer, but I have to start with the regulatory process. It can be deeply frustrating. I work with the rare disease families. It can be very frustrating when they know there's a potential therapy, and it's such a challenge to be able to move forward.
Yeah, the regulatory challenge is real, and the problem has been twofold. First, earlier generation gene therapies suffered from massive setbacks due to causing cancer. So that made the agencies incredibly conservative in the safety profile they would accept. Second, this is a new class of technologies, and new classes of technologies have undefined risks. Therefore, the regulatory agencies take, again, a very conservative approach, requiring massive amounts of backup data to push the IND through, push the new technology, new therapeutics into the pipeline. That path is slowly getting worn down as more and more therapies are coming on. But it is indeed very conservative. Contrast that to our situation with COVID vaccines. Prior to COVID, before the emergency use authorization, there was no mRNA therapies or vaccines in the clinic. In 9 months, we went through phase 1, 2, 3 clinical trials and global distribution. So indeed, regulatory capture is a problem. Secondary, and this is an R&D problem that Steve alluded to, a lot of these therapies are personalized. They are truly made from your cells for you. That's a huge overhead because no two-person cells are alike. For example, you're a leukemia patient, God forbid, but your immune system is exhausted from frontline chemo and radiotherapies. Now you have to go through extraction of these immune cells, which are immunodepleted and exhausted. These are technical terms, meaning they're crummy cells. They don't grow well. They're shipped off to a factory. That factory tries to multiply these cells massively. They may not grow well. 6 months later, they can do the gene editing, expand it further, send it back to you in the clinic. Meanwhile, you've died of cancer. This is the problem right now. This is why this costs millions of dollars. Now, the R&D part of this is people trying to develop universal donor cells. So if you can create immune cells that can be accepted by anybody in this room, despite of their immune profile, and not be rejected, because again, rejection causes cytokine storms and potential death. So rejection is very dangerous. But if you can make immune cells that can be used universally, that's the holy grail that'll bring the cost of goods down to, I think, a more manageable uh, um, level. But still, that's a new therapy. So that's going to be a whole different regulatory path. And God help us there.
And are those regulatory pathways pretty much universal around the world, or are there some areas easier to get things through?
Well, it's interesting. So I would say that Europe and the US do most of the regulatory path. In some cases, Europe is actually much more advanced in terms of the gene therapies in particular. They have the healthcare system that won't pay for any of them. So you can actually have the first clinical trials done in Europe, and then you have to have the FDA approval in the US to actually bring them to market. So it can be a very complex dynamic. So it's complicated, right? And I don't want to undermine. It's important. I have good friends in the FDA. I'm not trying to undermine. I actually think they've been given a charge to have zero risk when the risk of an action is not in their portfolio, right? And I think we really just need to come up with a process where the understanding the risk of an action is also balanced by the understandable risk of doing new therapies, right? And so I think we just need to reset actually around the humanity, which is the people in front of you that you're trying to develop the therapy. And I don't think we've quite done that yet. The FDA, Peter Marks, is actually trying to do this in a very meaningful way, but I don't think he has the regulatory path himself, legislative path to make the change. So I don't know what the answer is. I don't have a lot of optimism that there's going to be a legislative change, but I know that the scientists are really trying to find their way through this maze to make this happen.
But it's not going to be us. It's going to be the patient advocates.
Yeah, agreed.
It's not going to be us. It's going to be you. You're going to come. Somebody here or somebody has a rare disease that could be effectively treated by one of these therapies. And that's where you get involved. That's where you push a compassionate use policies and so on forward so that it can be done, maybe outside of the regular channels. And that's how you build the bridge to a regulatory pathway, usually through compassionate use or emergency use first.
Okay, next question for Michael Embry. What is the acceptable time frame for when an edit happens to when it's believed that the edit has no unintended consequences?
Let's talk about regulatory paths again.
That's the problem, right?
So if it's in, okay, so I would say that it's sort of where your sandbox is. So if you are pulling out and doing so-called ex vivo, so you're modifying B cells or you're modifying T cells, you can actually do a lot of testing of those cells in a dish before you put them back in a patient. And that's why a lot of ex vivo has been the most work. And to my knowledge, I know of there's very limited cases of adverse effects from CAR T cells, despite many thousands of patients that have been treated with that. So at the moment, the safety profile for that is pretty good. When it comes to in vivo gene therapy, there's actually not been that many experiments run. So I would say we don't really know. So I think that it's an important question. I don't know that we know. That's part of the FDA's challenge. They don't really know what that risk ratio is.
And kind of one question from my background, if it seems like a lot of the same problems that you're going through are similar to what autologous stem cell use and things like that can be reimplanted into the human N of one, modifications. Is that a true statement, or are there similarities?
Yeah. Yes. So I was at the University of Minnesota for many years before I moved to Mayo, and I don't know if you know, the University of Minnesota was the first place for organ transplants and first bone marrow transplants, and the first bone marrow transplants for leukemia. And there was a scientist who was the dean of research named Charlie Moldau, and he and I had this conversation, and he said that if the current rules in place, the University of Minnesota would never have done any of those, because the risk profile was entirely unknown. Are you going to put an artificial heart or a heart in a person, or putting bone? It was all entirely unknown. And for many years, bone marrow transplant, the survival rate was very low as you were doing all the work. And so it was him reflecting that that medicine had changed, but that the risk profile had dramatically changed to the point where his reflection was that the key signature science that was at the University of Minnesota, which still is a great place if you want to do bone marrow transplantation or the science, would never have happened. It was a very interesting, a very instructive conversation for somebody who was thinking about where the future of science was going in medicine.
Okay, and from Cyril, what are the potential risks of using genome editing to alter ecosystems or species conservation?
I mean, both risks and opportunities,
Mhm.
right? So we're altering ecosystems already. Drought-resistant crops are going to be the way we feed a hungry, growing planet. There is no doubt about it that CRISPR will play a significant role in reducing starvation across the world. Gene-edited plants are here and they are good. We can talk about that later if you want. So ecosystem change, somebody brought up plastic eating organisms. That's happening. University researchers here at UT Austin have some of the best plastic eating bacteria. It's amazing. I watch the videos. You sprinkle the stuff on a plastic bottle and it just dissolves over a couple of weeks. But the idea is to scale that up and do bioremediation of the ocean's plastics islands. So I don't know, risks or opportunities, you decide. Bioterraforming is happening. It's happening in negative ways right now. Maybe some point soon we can wrest control from forces of destruction and actually terraform in a good way.
I just offer, I like strawberries. I don't know about you, but it turns out strawberries are actually not very temperature tolerant. And so, if you want to be able to have strawberries, we're going to be able to add the strains to our ecosystem. I know those are artificial, but I think that, I would just offer that anything that's a clonally derived product, so navel oranges, seedless grapes, your coffee beans, your cocoa beans, those are all going to be plants where gene editing is going to save them from the pests and the environmental impact. I'm predicting that. I will take those bets, especially the chocolate.
And I should mention there's a lot of work now to bring back ancient or heirloom varieties and make the heirloom varieties as resistant. So for example, the monoculture of the Cavendish banana, right? So there's all of our bananas are derived from Cavendish, and the main feature of Cavendish is it's resistant to a blight that kills it. But it's monoculture now. And the way you get around that is you revive ancient varietals, but they don't have the adaptations that allow them to be farmed under modern climates, in modern conditions. So by splicing in the genes from Cavendish that made it so successful, but now so susceptible to this blight, we may now bring back and increase the diversity of our food supply by bringing in positive traits from monocultures into ancient species.
Okay, and for the last question, is Enrico Lynn Shiro here? Okay, great. Given what we know about the genes that allow some people to feel rested after only 6 hours of sleep, to grow muscle faster, less pain, et cetera, what are the ethics of making these changes in humans versus, I guess, the disease, curing a disease state? In other countries, as other countries start to do these changes in their population, how does this change the conversation around ethics and enhancements here?
So Ilya,
Easy question.
Yeah. I'll take because science fiction can be really inspiring. And when we launched the Genome Writers Guild, which was around the ethics of taking gene editing and bringing it into the real world, we had our first speaker, a keynote speaker, was David Brin. And I asked because he's a futurist, and I'm like, okay, you've already imagined. So, but I have bad news for you, Ilya. So in that prequel book, where it talks about the editing of the dolphins to make them be able to talk and be able to converse and be uplifted, David talks about the effect on the genome engineers in that world, and it doesn't go well for us. I'm just going to tell you. So I'm hoping that we can actually use this for the good so that Ilya and I are actually, not the villains of these stories, but are actually helping make the world a better place.
This is now me speaking as a futurist, not representing the views of my laboratory or the funders. But I have to think that the technology is, as you know very well, the technology is not that difficult, right? This is not like a Manhattan Project style thing. You could do this in your lab, in your home, with things you have in your home. So we've already made species with positive traits. And I cannot imagine that there aren't potentially non-state actors out there that aren't going to try this in the next 20 years or so. This is why the government is so interested in being able to trace whether something has been gene edited or not, or to lock down your own genome. And so then what happens? In projecting down 50, 100 years, I believe that if this does happen and you are able to make a subset of humans have positive eugenic traits, or even perhaps reduce the disease burden from cystic fibrosis or whatnot, then you may get into a situation where those humans will speciate. They will choose not to mix their genomes with those who haven't been edited. And we're going to have an event, much like Darwin's finches, where you're going to have specialization and speciation, but under our own control. Now, is that good or is that bad? That's above my pay grade. I don't get paid enough to answer such questions. But I think with any of these dual-use technologies, humanity has rarely shied away from the very obvious risks that they possess. Think about nuclear power and so on, right? We're at the brink of I don't know how many seconds are we from nuclear destruction according to that atomic clock. So we've never shied away from dual-use technologies, and I don't see why this will be any different.
All right, thank you all. Thank you, Enrique, for the question. Make sure you find us to get the badge.
All right, thank the speakers. Thank you guys very much.